Okay, so we'll get started. Good to see some faces which had disappeared. The goal for today is to do something really new, um, which is CRISPRing the genome. Why I'm saying it's new is because this field is not very old. This field is, it is old in the sense that we have been doing gene editing for a while, but the way we have been doing it is very new in the last, I would say, 15 years. Okay. So before we get started, some logistics to take care of. So initially I had planned 12 assignments for a variety of reasons, including some assignments took longer. We had some break in between. There will only be, I think, 8 assignments in total. Your lowest 2 will be dropped. So there will be an NSEM examination, but it won't be like a full-on traditional NSEM examination in the sense that it will be just for you a venue to just come and have fun doing some sort of just answering very simple questions, right? It won't be mathematically as heavy as the mid-sem was. It will be a simple quiz. You can think of it as a simple in-class quiz which is based on material which is taught after mid-sem. So this will involve just interpreting plots, suggesting analysis, diagnosing issues. So you have to just act as if you are Sherlock. You're given an issue and you're trying to figure out what that particular, how will you solve it, right? So for example, the stuff that we're going to cover today will have some technologies being discussed. So I could give you an example where I'll just ask you that, okay, this is the scenario, what kind of technology will you use, right? So very simple, does not require, first of all, will not require calculations. It will be just very sort of interactive, just require you to think from first principles. That's what it would be. I don't expect it to take 3 hours. I think it's probably a half an hour or a 1 hour thing. This will also be the venue for submitting your posters. By posters, I mean you will print it on your own and we'll figure out the funding issue in just in maybe by the next class. But the PDF of the poster and the report of the poster needs to be submitted in class Friday, 22nd November, right? So this is kind of the deadline that we already had. Nothing changes. The exam is on that last day. Nothing changes practically for all the reasons. The only thing is what you would have been doing from your room, you will just come here and do it, right? So this is almost like doing a very small in-person assignment. That's what it is, okay? So don't get alarmed, don't freak out. Nothing has changed majorly. This is just another assignment submission happening in class, basically, okay? So lectures for 1st, 6th and 9th November will be pre-recorded, so there will be no class. There will be no class here. We will not be meeting in person. This is because I'm traveling for that entire 10 days, so I'll not be able to hold them in person. Okay. Any concerns? Any questions? Okay. Okay, so then let's get started, and by asking a very simple question that if you want to understand the function of a gene, what kind of analysis would you do? How do people study the function of a gene actually? So we know there is transcriptome available. We know where these genes are, but how do people profile where are genes located? So how would you, if you wanted to understand what is a function being carried out by a gene, what would you do? Knockout, okay. Where was it coming from? Knockout, okay. So what would knockout tell you? If those genes is missing, then how things are going. Okay, fair enough. Any other ways to study a function of a gene? Okay. And what does that achieve? Okay, but how would you study what would you measure? What is the, so you knocked out the gene, but what do you measure then? Okay, difference from normal behavior. So you need some measurement, right? You essentially require some sort of a measurement. If you do any any sort of functional testing will require you to have some sort of a measurement, right? So people have been thinking about this for a while, and these experiments that you're suggesting, knockout or overexpressing it or expressing it in a different organism, these are costlier experiments, right? So that's the right way to test it. But given that we have been studying these genes for almost 20, 25 years, or maybe actually more than that, like almost 70, maybe 100 years, 100 plus years, right? We do know a lot about genes. We know about certain genes, what function they carry out. So this has been cataloged in these databases. So the easiest thing to do is to use these databases where somebody has already chalked out what a particular gene's function is, right? So this started from the protein world, particularly the enzyme classification system, or the EC system, which was one of the first comprehensive protein classification system, right? So what it does is it takes all your enzymes and classifies them into 6 categories: ligases, isomerases, lysases, hydrolases, transferases, and oxireductases. Okay, so EC1 to EC6, these are 6 different classes. All the possible enzymes that we have been cataloged in humans can be put into one of these 6 categories. And this is a very easy way to catalog or to figure out what kind of function would a particular enzyme be carrying out, right? So this was developed like very, very long ago, even before we started sequencing or doing genome sequencing. And this is a great system for proteins or enzymes in general. The same need was felt when we started to think about genes. When we started to catalog genes, how do we think about what is the function so genes are producing proteins, so enzymes are a special category. What kind of function are these genes carrying on? So that was initially a system was designed for Drosophila, because Drosophila or fruit fly is a very commonly used model organism for understanding genes, right? So people were doing all sorts of genetic testing on Drosophila, and the thing that they want to understand, okay, if somebody, there are like 100 different labs in this world who are studying Drosophila along with me, and 50 of them have figured out that one particular gene carries out one particular function, right? So for example, one gene carries out this patterning thing, then I should be able to leverage that. I should be able to leverage what somebody has studied for a particular gene. They designed what's called an ontology system. So an ontology is just a categorization in the form of some terms. These are a common language that everybody understands in some way. So some effort was put into categorizing the function, or like it's not technically the function, but what is sort of the association, or what is the terminology that is associated with each gene. So this standardized terminology, which is called gene ontology, is a way to describe proteins or a gene's molecular function, its the association with its biological process, or which location of the cell is it acting, right? So we performed gene ontology analysis in the previous assignment or previous hands-on, and I guess if you have done it, you would have also come across it in the assignment. The goal for that gene ontology analysis is to like do a poor man's or poor woman's way of figuring out what is the function that a certain set of genes is carrying out. Okay, so we'll just see in a bit how that thing is carried out. But overall, what is gene ontology? It's a categorization of the gene in different categories. So for example, I could start from a very specific category, which is beta amylase activity. So one gene could be part of this. This is an ontology term, which itself has, so this is the parent and this has different children. As you go along so it's a directed acyclic graph, it's a DAG, which tells you as you go down the hierarchy, it tells you how specific a particular function or a particular term is, right? So something like, let's say, DNA binding would be a very generic term. This would be something which every transcription factor would share. But if a particular transcription factor is carrying out, let's say, repression, then there'll be a further category below it, which will tell you that it is specifically related to. All of these are DNA binding, transcription factors are DNA binding, but then it's going to carry out a very specific function. So we have a system for cataloging what a gene's function is. So we have 23,000 or 20,000 protein coding genes. One gene can be part of multiple different ontology terms, which means that it could be associated with, let's say, RNA, DNA, or RNA transcription, it could also be related to RNA or protein translation and so on. So it could be shared by one gene, could be part of multiple categories. Similarly, multiple ontology terms can have, one ontology term has multiple different genes associated with it. And the way you define a particular ontology is this kind of a directed acyclic graph, that one ontology term is associated or is a function of B, which is indeed part of C. So for example, your organelle membrane, this could be a function, this could be an ontology term. This is part of a very generic term called organelle. Okay, so once we, the reason we want to sort of look at this is to understand what the function, likely the function of that particular set of genes is. So what we did in the previous hands-on was we looked at a number of differentially expressed genes that were different in microglia cluster between Alzheimer's patients or Alzheimer's mouse and normal mouse. Now there are like 50, 100, 1500 different genes, right? But what does it really mean? What is the function that these genes are carrying out? If you have access to a database which has the terms for each of these genes cataloged, you can ask which of this term, given I have a list of 500 or 1000 genes, which of these terms keep appearing again and again, again and again. And you can check for this statistically by doing what's called a hypergeometric test. We did not explicitly do a hypergeometric test, but it's not a very difficult test to understand. All you're looking for is, given you have a set of, let's say 20,000 genes in the human genome database which have annotations. So each of them is associated with a particular gene ontology category. You come up with, let's say, 1000 differentially expressed genes. So your M is 1000, which is the number of genes that are annotated in your data set. N is the number of, is the size of the list of genes of interest, right? So N here is the number of genes that are annotated, which means that if I take the 100,000, if I take like 500 genes, this number is 500. Now I'm asking which among these 500 genes that have an ontology category, which category keeps appearing again and again, right? So for each of these categories, I can list how many number of genes were there in that original category and how many of them overlap with my gene of interest. So N is the size of the list of genes of interest. So I start with a particular set for the genes, and N is the number of genes within this list which have an overlap with my genes of interest. So let's say you have 500 genes which were differentially expressed. You start from that number 500. You come back to check which of these genes, given the different categories that you have, let's say you have 10 different categories. For each of those 10 categories, how many number of genes that are there in the original category and how many number of genes are there in your category, how many of them overlap, right? And you can do a test, a statistical test based on that number, whether that overlap is significant or not. And you calculate a p-value using a hypergeometric test here. So this is what we did in the previous session. So this is taken from that session. So on this, I'm plotting the p-value, right? So the p-value here is when I started with my original gene list of differentially expressed genes, let's say top 100, top 200 gene list, around 3 genes. So this is the number of genes that overlap with a particular category, and this is that category. So here you probably cannot see it there, but it says positive regulation of endocytes, right? So this is a particular gene ontology category. Of the 300 genes, of the 100 genes, excuse me, 3 of these genes have an overlap with this particular category, and this is the corresponding p-value, which is that what is the chance of, or what is sort of the observation, what is the p-value associated with this overlap coming because of random chance, right? Very loosely random chance. It's not exactly the right definition of p-value, but overall, what is the chance of observing something as extreme as this, which is 3 genes overlapping with this category, and that p-value is low, which means that this term is appearing again and again over the genes that you identified to be differentially expressed. That's what we do. So this is a poor man's way of figuring out what likely function your different genes are doing. But the fundamental question that we asked is, how are these functions themselves derived? Like somebody would have to spend their effort figuring out that there is a term which is related to positive regulation of endocytosis or positive regulation of gene expression or positive regulation of amyloid beta synthesis, right? How is that determined? That is going to be our focus today. Is the general problem clear?
How are we calculating that p-value? I mean, I see that for different
ontology categories, yeah. These are the different ontology categories.
Different categories, but even when they have the same gene count, the p-values are different.
That's right. So that's a good observation, right? So the reason these p-values are different is because the categories have different number of genes to begin with, right? So each category can have 5, 10, 15 or even 1,000 genes associated with it.
By category you mean that
ontology category, right? So 3 is the number of genes that overlap with this, but all of these categories themselves, for example, this could have had, let's say, 100 genes, this could have had 500 genes, this could have had 200 genes. So that's why that base number of overlap is different. So that's why your M here is going to be different. So that's how your p-values are going to be.
What are we looking at the overlap with? So this for overlap we need two things, right? The first thing is the ontology term which is on that.
Let's do an example actually. It's a good question because I think I need to. Okay, yeah. So let's say you have a term A. It does some function, okay? Whatever function it does, and it has all these genes are associated with this function. Okay. Then there is another term.
And one gene can be associated with many.
Yeah. Let's take that example, right? So this is G1 and G3, and then there is some term C that has, let's say, just G3. Okay. I mean, maybe G2 also. Now I do a differential expression analysis and I find out that I have gene 1, gene 3, gene 5 and some gene 10 in my list. Okay, this is my input list or differentially expressed genes, DEGs. Okay. Now what I do is I go for each of these categories. So if I go for this category, I get one overlap. If I go for this category, sorry, I get two overlaps. Here also I get two overlaps. Here I just get one overlap. Okay. So on the x-axis, what I'm plotting is the number of genes. So this will be 2 here, this will be 1 here, this will be 1 here. Okay. Now in terms of the p-value, what you're calculating is what is the overlap.
It will be 2 for term B.
Oh, sorry, yes. Yeah. So what I'm calculating for the overlap is, what I'm calculating for the p-value is, given your, let's say you had 15 genes in your entire genome which have some category associated with it. Out of those 15, 3 are part of this, right? So that is you're dividing it into 3 bins, and out of these 3, 2 of them overlap. So what is the associated p-value associated with that? That's the kind of function that we are doing. Is that clear now?
What is this set of differential expressions?
So this gene is.
Anything.
It could be anything, right? So let's say I take my liver, I take my liver and I take somebody else's liver, and I'm just interested in what is different between these two, right? So I identify that there are 10 genes which are differentially expressed. But now I want to understand, like, these are just genes, right? Like, I just have no idea what these genes are doing. So my goal is to get some idea of what is the likely function or the process that is associated with these genes. So the way I'm level, so ideally what you would all like to do, you take this DEG list and you want to figure out what is the function of each one of them, right? Like in an ideal world, you would say that, okay, let me knock this out, right? So somebody here said like you should knock this out. So I'll knock out, I'll measure what happened, I'll do the same thing, I'll do a knockout, I'll measure what happened and so on. And that will take ages and like such an extremely expensive experiment to do. So you said, okay, let me not do this because I'm not the first one to study gene 1, gene 3, gene 5. Somebody has done some jobs for it, and what I know is this is part of A and B. This is part of A and B, and I don't know what these two are part of. But it seems A and B are occurring again and again, right? So it's very likely that whatever this A and B is, that is the function that they are carrying out in this particular context. So that is what your gene ontology is.
But if A and B already had a lot of genes associated with it, then we
very good point. Yes.
We won't consider this unlikely?
Yeah, so that's where the p-value helps to some extent, but there is one sort of a good catch which you're kind of highlighting is, people like to study, for example, brain or cancer, people like to study a lot. So some of these terms have a lot of genes associated with it, right? So let's say if a term A had like 1,000 genes, right? And if you just do differential expression with 100 genes, in a lot of likelihood it will appear to be significant, which means that you will find it just because there are so many genes, right? So like the extreme example would be that all the 10,000 genes are part of term A. So whatever you do, it will turn out to be significant. So you have to be a bit careful about like interpreting and over-interpreting these terms. But from a very first pass, it gives you some brief idea of what these genes are doing, but obviously take it with a grain of salt.
How about we add another axis which tells how much that gene affects that term?
So how would you do that? Yeah. So how would you do that?
I mean, assume like this term represents some pathway, and then one of these genes.
That's a very good point. You could do that. Yeah. So you would look for the exclusivity of a gene to a particular pathway, right?
Not even exclusivity, the important significance. Suppose gene 1 is absolutely essential for that pathway to function.
Yeah, that's a hard experiment. That's a hard experiment, right? The reason biology is so hard, and the reason it's so hard is because none of this acts in an independent way. There is an interaction built here, right? So if I just do a knockout of this particular gene, it will not mean that only that gene's effect will come up. There is what is called epistatic effect, which means that, so if I knock out gene, let's say I get effect A. If I knock out gene 2, I get effect B. And let's say if I knock out gene 1 and gene 2, what would you expect in terms of relationship? What kind of effect would you see?
Probably worse than no.
Yeah, so it is a combination of both, right? It's something which is very, very hard sometimes to even figure out, and the trickiest part is actually it's not even sometimes this, like it's a hard relationship to figure out, and that's one of the reasons biology is so hard, right? None of these things acts in independence. This could have been part of one pathway, it could have been part of a different pathway, but these pathways somehow interact because there is some protein which acts in between, which causes them to sort of interact with each other. That's the reason biology is so hard. Okay,
so do people study these kinds of effects?
Yeah, they do. So that's one of the technologies that we're going to talk about. That's the aim towards going through this stuff, right? So what is the most ideal experiment can you do if you want to study this? If you want to study all sorts of interaction, what would you like to do? Just design an as expensive an experiment that you can. What kind of experiment would you design?
Let's say I want to study gene
All 20,000 genes. I want to study all 20,000 genes.
Monitor all kinds of biological markers.
Yeah, so you will basically do some sort of perturbation to knock out each one of them, right? Each one of them, one by one, keep rest of this remaining constant, and then all possible,
all combinations.
All combinations, right? That would be the most ideal experiment to figure out everything. However, very, very expensive to do. We are inching closer towards it, but not there yet. You had a question? Okay, so we already talked about some of these ideas, right? So we want to study the impact of a gene. How do you do it? One of the things that you could do is you could do what's called a knockout experiment. And this knockout is a very generic term. There are different ways to knock out a particular gene. So the earliest methods for knocking out a gene was purely based on what we understand about genetics. We did not explicitly cover this particular thing in the class, but it's also very, very easy to understand. What you do is you have a particular chromosomal DNA. You want to knock out this particular gene. You go to its gene's vicinity, to its 5' region, 3' region. You synthesize a particular vector. So you take a particular E. coli or any sort of other organism. You can engineer them very, very fast. You design these sequences which are complementary to your original gene of interest, and just put this along with your rest of the system, right? What this will eventually happen is there is something which is called homologous recombination, which essentially just means that once these two start to complement somewhat, there will be some crossing over. So if I start from this sequence, this is my gene of interest, and I start from a template here. At some point when replication begins to happen, what will happen is I'm adding I'm adding a new sequence here, which is this. This is very, very different from my gene of interest. At some point, they will hybridize together, and your new copies will have something like this, and some other permutation combinations of it. So it could also involve. Okay. Essentially, what you've done is you've broken apart the open reading frame, or the reading frame in which the translation was going to happen, right? That's the essential thing that is happening here, and that will disrupt the expression of that particular gene. It will either not get synthesized at all, or you will end up making something else. Generally, you don't end up making anything because you end up also incorporating a stop codon in this case, right? So you have this is your start, and this was your stop. Because of this, you will now have a stop appear prematurely, and shorter proteins can be degraded very, very fast. So this will end up disrupting the gene. Okay. You can also use something which we talked about, siRNAs or RNA interference. The idea is that we have already a mechanism in place which can play around with the viral DNA. So if any virus infects, or based on double-stranded RNA, if there is a double-stranded RNA, this particular mechanism evolved to undo any sort of reaction that can come because you end up ingesting an RNA, or the cell ends up ingesting the RNA. And briefly, what happens is once you ingest a double-stranded RNA, your dicer is a protein. It will come, it will perform an endonuclease reaction, it will chop this off, and this will form very short fragments. This is siRNA, right? So this is first level of degradation or killing the virus RNA that happens. And at some point this will get degraded, it will form a single strand RNA, and if this single strand RNA can now reverse complement to some copy of your mRNA, okay? So if you if you can somehow inject a double-stranded RNA such that some of its sequences can reverse complement to your mRNA of interest, you can use this, which is called RNA interference. This is basically your siRNAs. This will end up causing the mRNA to be silenced, because once it when it hybridizes to your mRNA copy of interest, there's a particular protein called Argonaut, Argonaut, which is able to recognize this kind of double bonding happening at a particular region. It will go there, cut it. This will end up again being premature, and it will end up getting decayed. Okay, this is also still used. Actually, this is still very, very efficient this is not bad in terms of achieving.
Does it inhibit the function of other genes?
Good question. It can actually interfere, but this is more specific than some of the other things that we'll be seeing.
It's like it will change like the conformation.
Confirmation for the mRNA, or
Yeah.
so it will actually once you are here, right? The Argonaut machinery will actually come and recognize this and just totally decay it. It will totally end up chopping this off. So you'll end up even not synthesizing the protein.
So you'll have two different RNA strands.
Yeah, but eventually you will have, exactly. So you will cut this off, but there is something called NMD mechanism in the cell, which is always sort of surveilling the cell for these short RNAs. It will end up chewing up all these short fragments, which are not stable. So that's why mRNA can decay very, very fast, because there is a mechanism that can clear it up as well.
I mean, what about the other genes?
What about the other genes? Will it affect the other genes? That's what you're asking?
Yes.
So it can. It is because sometimes this mechanism does not need to be an exact hybridization. So there can be some of what is called off-target effects. They're not as bad as it is there in other technologies. So there could be off-target effects with RNAi as well, but they're not as pronounced. It also actually depends on how you're synthesizing your RNA, how specific is the sequence of your siRNAs, because it has to, if this particular sequence is only specific here and very, very specific, then you will not see a lot of off-target effects. Okay, then you can come to one of the other suggestions here, which is you can start to overexpress a particular gene. So the way this is done is what's called transfection. You start from a particular set of cells, you would start from a particular gene of interest, which is your DNA, and you will clone it into a vector, which is basically you're again taking somebody else's machinery. Now this machinery is so special, so this is what you want to clone, this is what you want to make copies of. You take this particular sequence, this is your protein, and you implant it into a particular area where this promoter is highly active. So if this promoter is highly active, what it will end up doing is, once you transfect this into a particular organism, this will become active and it will start synthesizing multiple copies of the RNA. So if you insert, so we actually generally insert the cDNA of this particular gene of interest onto these cells, such that this viral vector copy has a very active promoter, so it will make a lot of copies of this cDNA. But before we see what exactly does that, any reasons why we should use cDNA and not genomic DNA? Genomic will be more stable. Which one will you prefer actually, cDNA or genomic DNA? You have access to a vector. So the vector, what it does is it has this promoter sequence. You can implant anything here, and the moment it gets implanted, your machinery will come into picture, which is that your RNA pol will come and like start to synthesize and so on. Yeah. Okay, good, very good. Okay. Right, so there are no introns here, right? So if there are no introns, it actually helps you. It's very shorter, it's easy to transplant, it's easy to sort of manipulate in a test tube. It's much easier to handle. Introns are very long sometimes. You did an assignment, I guess, where you saw the size distribution of introns. So that's why we use cDNA, and this particular highly active promoter will direct the expression of this particular clone gene in these mouse cells. Then you can understand. So you have to still understand the phenotype, right? So fine, I have taken the gene, I've implanted it in the mouse liver or wherever I want along with this vector. Now it's doing some overexpression. I will verify whether it is happening or not. And then I will measure in some ways what is the impact that this particular thing has had on the mouse as compared to a normal wild type mouse, right? This could involve doing another RNA-seq experiment to figure out what other things changed. This could involve just observing the mouse. Are they, for example, people who do behavioral research will implant a new gene and then try to understand whether is this mouse more active now, is this mouse more lazier now, is this mouse going to be more obese now. So there is all sorts of phenotypic, so that's what the phenotype is, right? So the phenotype is something, a very generic term, depends on what exactly you want to but it should be something which you can quantify in terms of the changes that are happening, right? So it could be at the molecular level, it could be a molecular phenotype, or it could be a behavioral or external phenotype. Yeah.
I just had a confusion.
Yeah.
Is it always necessary that the function that a certain gene shows in organism A will also be replicated in the model organism?
Yeah, I think I made a joke once, which is a lot of papers come out on Twitter which says that X gene causes cancer, but they forget that it's in mouse, right? So that's very point on. Like what it does in other organism is hard to figure out. We cannot be 100% sure, but also these experiments are costlier or like very tricky to do in human, right? So you have to find a human who is ready to donate himself or herself if you want to do this in vivo. So that's why we do it in mouse. There are some ways to sort of verify this, which is if it does work or if it shows some kind of a behavior in the mouse, you could come back and try to see at different levels how similar are the sequences, basically, right? Like, is the remaining, so I only implanted this particular sequence, but are the remaining other sequences more or less also consistent between human and mouse? So that is one sort of like sanity check that you could do, but the only way to sort of figure this out is to do this not in mouse, but like closer organisms like macaque, chimpanzee. So that's why the clinical trials begin there first. First it used to be dogs, but then somebody realized that dogs are not a good model organism. Mouse is a good model organism because everybody can do it in the lab, but like the testing when it comes, you need to do it in primates. Primates are chimpanzees, macaque, bonobo, and all sorts sort of like closer neighbors to human, basically. Again, so that you minimize the effect of this thing. of any other external factors.
But any other experimental method that has been, or any other experimental strategy that has been devised to minimize this kind of thing?
So yes, you could do this also in cell lines which are immortalized. So for example, I'm doing this in vivo in the mouse, which is, you could argue whether that's better than doing it in vitro. So people takes these cell lines. For example, you would have heard of this cell line called HeLa cells, right? Henrietta Lacks, she had some form of cancer, and for some reason the cells never stopped growing. This cell was cloned, I think, or separated from her in 1930s and still grows, like you can grow them in colony. And now there are like multiple different cell lines like that, which are human, right? So these are coming from human samples, with the caveat that these are diseased cell lines, right? So there is something wrong obviously with them. You can also do these experiments in cell lines, but again, those will be in vitro. So yeah, there is no like middle ground, and like there's no absolute middle ground in terms of figuring out the best of in vivo. So this is in vivo, right? Like the mouse is alive and still able to function and so on. Those are immortalized cell lines, so you'll have to come up with some sort of screening mechanism to figure out whether it is expressing the phenotype or not. But yeah, otherwise there is no way to do this. People are working on this. For example, there's a lab, George Church, you would have heard of. He's trying to revive mammoth by sort of, so if we can revive mammoth in some ways, we can revive, like we can clone entire human bodies at some point. Again, this is all very speculative and I think very, very early days, so on. So if that happens, then yeah, maybe we should use those as but yeah, a lot of ethical sort of stuff in ground to figure out. Okay, so then comes our genome editing, right? So this is very, very new, like 15 years probably, in terms of what we'll see the details in just a bit. But even so, you'll realize that some of these slides will get extremely technical. But what I want you to remember, if you want to understand genome editing, is this particular figure here, right? So the idea behind genome editing is again very much driven by what we studied in molecular biology. So we want to make the gene to be non-functional in some way, right? We want the gene to be edited such that it does not produce the protein, or it is not making enough number of the copies of that particular protein. So how do we do it? The way to do this is to perform some sort of editing so that the final edited sequence will not have the same properties of your original sequences, because now if you have a new sequence, it will not be the same protein, or it could not be even synthesizing that protein. So how is it done? The general principle is you take this double-stranded DNA, and you figure out at certain place where you want to make that edit, right? So this is something which you figure out, which particular part of the genome you want to edit. You design a specific sequence against it, which is called a guide RNA, and we'll see why it's called guide and why it's called like that in just a bit. Once the guide RNA comes here, it is not coming by itself. You also make the cells express a particular protein which is called Cas9. It's an endonuclease. So endonuclease will chew off any fragments which are sort of not protected in a way, right? So you take this endonuclease will make a double strand cut here, right? So the reason right now it's going to make this cut is because there is some sort of misalignment that has happened here, right? So the guide, there was already a double stranded cut here, there was already a double-stranded DNA here, the guide RNA came, started to hybridize, and this has now started to unfold. That activates your Cas9 endonuclease system, which will come and make this double-strand cut, right? It is all basically driven by that at some point you need to repair this cut. So once you make this cut, so the endonuclease is such that it is kind of attached to this guide RNA. And once you attach that entire mechanism here, once it cross hybridizes, it will start to make cuts in the nearby regions. So once it makes these cuts, the repair machinery will get activated. The repair machinery is very, very fast, right? So whenever you make these kind of cuts, the DNA repair machinery will come and will start to heal this, right, in a way. The way this is healed can be slightly different. You can do either homologous end joining or non-homologous end joining. We'll see an example in just a bit, but in most cases it's non-homologous end joining, which means that You just heal this part, you just heal this part, there is no crossing over that happens. And because of this, what happens sometimes is there are two things that can happen. You can end up making a deletion here, so you can end up sort of synthesizing these two different, you can end up joining these after having made a deletion here, or if there was some other sequence that you had imparted, it can get incorporated here, which would lead to an insertion coming up at this particular place, right? So this non-homologous end joining mechanism is not like the most, this is generally not what happens. Like the copying mechanism mostly involves some sort of homologous end joining because that's more exact. Non-homologous end joining is error prone, so it will result in a short insertion or deletion, which will end up disrupting the open reading frame. So it will end up either imparting a stop codon onto it, or it will end up imparting a particular sequence onto it, which will lead to the protein not being synthesized in exactly the same way. Yeah.
Why at the first place a guide RNA is, insert? I mean, what is destabilizing this structure so that a guide RNA can
Yeah, so what is destabilizing this, which is allowing you the guide RNA to hybridize, right? So in terms of this mechanism, what exactly happens is at this point, whenever you impart, this is very similar to what happens generally. Whenever there is a short sequence which can hybridize, it will just go and start to hybridize it. It will be in the vicinity. Just because you also have an endonuclease present, that is so nothing would have happened if you did not add the Cas9, which is that things will hybridize, but nothing really will happen. But at this point, this is just your pure chemistry acting in a way, right? This is just hybridization happening because it is bound to happen because it's in the vicinity of it. But just because you have the endonuclease, it will end up cutting it. So that's the difference between, like, this keeps on happening in the genome all the time. There is always hybridization happening in some way. This is just a way for it to sort of stabilize in a way. Okay. Then comes this idea that one of the questions that Dhananjay asked, right, which is how will you make it specific, right? So what you're doing here is you're synthesizing a guide RNA, and this is designed so that it can go and cross hybridize to, it's not as simple as it is in this figure here, but we'll come on to the details in just a bit. But if you are designing a guide RNA, there is no reason that it will only hybridize to a particular site to which you design it against. You can also hybridize to certain other sites where there is not a perfect match, but it can still do a matching there. Yeah. The structure is what? More stable. Which structure is more stable? Good question. My guess is this is all an intermediate process, right? Like this structure, I don't think is the most stable structure, but just because this sequence is attached to the endonuclease, that reaction takes over pretty quick, and it ends up starting doing the chopping off operation right here itself.
So you need to less guide RNA and access for this process.
Yeah, good question. So you need so when we start to do this at a single cell level, you need enough guide RNA to be expressed for this to actually kick in. Yes. So that does require some sort of optimization. What is the right amount of guide RNA where you will start, this mechanism will have to start to take over. So we'll see that in just a bit. Okay, so this could have off-target effects, and one of the reasons we are sort of still debating of how therapeutically feasible it is. By the way, it's kind of, we are getting very close to it being therapeutically feasible, particularly if you go back to the first or the second or the third lecture of the class, which was, I talked about sickle cell anemia. FDA approved therapy for treating sickle cell anemia using very similar principles, not exactly the same principles, but very, very similar principles. Okay. So how was it discovered? It was actually discovered in prokaryotes, right? So this, you would have heard of CRISPR-Cas9, right? So the CRISPR is, so this is the principle of genome editing. This is the principle at this point, that how can you do it? Like, we have not talked about how exactly it is done. Now we're going to talk about how was it exactly discovered and how does it exactly work. So it was discovered of all things in prokaryotes, right? So we are doing this in eukaryotes, but it was discovered in prokaryotes. Prokaryotes have this thing called CRISPR, which are basically clustered, regularly interspaced short repeats or short palindromic repeats. So CRISPR is a short form for clustered repeats, essentially. What essentially, this mechanism has evolved is very interesting, right? So prokaryotes, which are generally bacteria, are prone to be invaded by viruses or bacteriophages. This is a bacteriophage. This bacteriophage, what it does is obviously by itself is nothing, right? Because it doesn't have its own replication mechanism. So it will infect the DNA, infect the bacteria, and it will inject its DNA, right? So sometimes these are mostly retroviruses and so on. They will inject their RNA, and then eventually that will get converted to DNA and so on. But you have assume you have some sort of infection that is happening, which is a double-stranded DNA, and that gets part of is now injected. Now what will happen is the bacteria has to fight this, right? The bacteria needs to figure out how can I get rid of this as quickly as I can. So it has evolved a mechanism to do this. And how does it do it? Is by keeping literally a record of what previous infections has it seen. It does this by forming a CRISPR array, right? So CRISPR array is nothing but short fragments of DNA taken from the virus's DNA, and those are being arranged onto this particular array, right? So this array is made up of one fragment of a virus followed by a repeat sequence. This is called PAM. We'll see this again in just a bit. What is exactly PAM? So this is sequence that is taken from the DNA of the virus. How did it eventually synthesizes? Is because the bacteria has the Cas2 and the Cas3 proteins, which can go and perform this endonuclease digestion, right? So they will go take the virus DNA, chop it off, and once it chops it off, it will start to synthesize that, okay, I need to keep a memory of this particular infection. Because the next time this happens, I want to be able to do this really quick. Before anything happens, I should be able to recognize it and just chop it off really, really quick. The way this does it is it creates a copy of one of the fragments of the DNA coming from the virus into this CRISPR repeat. It will perform a transcription, and now this transcription becomes what will become essentially your guide RNA, right? So this is, we'll see this again in a bit. But the next time the infection happens, it can start to, at this point we have only focused again on the DNA, right? And if you were paying attention to the siRNA story, you would have realized that just killing the DNA is not sufficient, because sometimes by the time it would have already transcribed into the RNA, and you want to chop off the RNA as well, right? So it will start to, so the second layer of it involves synthesizing these guide RNAs. These guide RNAs will now be ready, right? So they will get attached to these Cas proteins, and the next moment any viral DNA arrives, it will start to start the chopping off operation or the killing operation there itself, right? So it doesn't need to repeat this cycle. It keeps track of this array. It keeps track of which viral DNAs have existed in the past, and it uses that mechanism to attack any foreign sort of.
Same chopping mechanism as it was in the
same chopping mechanism, very, very similar chopping mechanism. Just the Cas proteins are slightly different. So this is Cas2, Cas3, but that's Cas9, but the chopping mechanism is exactly similar or same. Okay, so what does it look like? So what are the different sequences here? So you have the first process is of spacer so whenever you have a particular infection happening, you get Cas1 and Cas2 proteins. They will chop this off and make it part of your DNA sequence or the spacer sequence here. Next step that would happen is you have these spacer sequences. These are spacers. So these are also called proto-spacers. These are separated from each other by what's called a repeat. These are also called PAMs, proto-spacer adjacent motifs. The next step that happens is of RNA biogenesis. So this can be transcribed, and then this becomes your guide RNA, which is attached to your Cas protein. Now this is ready to attack any new DNA that comes in, right? So this is sort of your surveillance mechanism or a surveillance complex. Anytime you have a new infection coming on, it will use the sequence to directly attack it, right? So some of these DNA fragments are shared between different viruses. Again, viruses keep mutating, but some of these, if this sequence of the guide RNA happens to match a new infection, that virus will be killed directly there. If not, it will get chopped off and finally get incorporated into the spacer array. Like that's the mechanism that the bacteria has evolved to fight the infection coming from bacteriophages. That is the principle that went on to win the Nobel Prize in Chemistry in 2020 to Jennifer Doudna and Emmanuelle Charpentier. I think I cannot pronounce her last name. She's French. I mean, the name is French. So in terms of what is happening, essentially, we've already described this, but this is taken from the Nobel press release. So what they discovered is they were not doing genome, they were not interested in genome editing. Like, so these people were interested in understanding how does the virus or how does the bacteria evolve to fight all sorts of viral infections, right? So these are basic scientists. They were interested in, they still carry out, part of their research lab still carry out basic research, which is like, they don't care about like what is gene therapy and how you can use this and so on. All they cared about is how does the bacteria evolve. That's all they cared about. And this mechanism, once they discovered in prokaryotes, they realized that if we can somehow emulate this in prokaryotes, that will be a game changer. And that's what was done in the 2012-2014 paper, which I forgot to link. But the general idea of the genetic scissors is explained in this figure, which is, if you can start from a guide RNA sequence, you start from a Cas protein. So Cas9 protein, for example, is there. You can engineer the cells, or you can impart the cells to manufacture Cas9 proteins in eukaryotes. This Cas9 protein, when attached to the guide RNA, will become your endonuclease machinery. The guide RNA can reverse complement, can hybridize to a particular sequence in the DNA which you want to chop out or which you want to remove. Once this mechanism starts to kick in, there are two possibilities. One is that you will end up doing a non-homologous end repair. So this is non-homologous in the sense that once you make this edit, this gets chopped out. The repair machinery will come and start to sew this back together. And in the process, it will do some sort of this is not an exact mechanism. So NHEJ, or non-homologous end joining, will end up incorporating errors here. And because you have ended up deleting or inserting new DNA, making these errors while repairing this process, you will end up manufacturing or synthesizing a wrong protein, or a protein which is not of interest, basically, right? Or maybe not, it's not even a protein at all now, because the stop codon happens to be here. So you have essentially ended up knocking out that particular region of the genome. So this is primarily focused on protein coding regions, but we can do this also at the non-coding regions. You can also add a new template DNA, so that once this non-homologous end joining repair machinery comes into picture, rather than incorporating some random DNA, it can incorporate a special strand of the DNA, and that becomes your insertion. So you can insert a new sequence as well, and that's also sometimes leveraged in the field, but most people use it mostly for knocking out genes in particular. Okay, so in prokaryotes, this is just a sort of a repetition to give you a flavor of exactly what is happening. So we have a parental array. This is what happens in prokaryotes. There's a leader, there's a repeat, there is a spacer, right? So these spacer sequences come from our fragments of the previous infection of the virus. They get incorporated and keep getting incorporated as an all new phage keeps attacking the bacteria. The next time this infection happens, so if there is a reinfection, this gets transcribed, forms part of the Cas complex. Cas complex can come directly chop off the entire viral DNA before it starts to make anything in the cell at all.
Does it pick random sequences from the thing?
No, so that's a good question. I actually don't know in terms of, I think it figures out, in some ways it tries to figure out like what is that common sequence. So there are certain properties. The properties it has to follow is that it has to be, in some ways it's kind of specific, and I actually don't know how
it's not a critical section of the phage, then it will still be right? So the bacteria needs to ensure that it's attacking a critical section.
So it has to attack the critical section. The tricky part is like these bacteria will, the viruses will keep evolving in a way, right? So it has to attack, ideally for example, it has to, if let's say the coronavirus has to attack if it were to attack the bacteria, it will figure out what is the most common, it has to figure out what is the most common sequence which does not get mutated, and that's a technically challenging problem, I think, for the bacteria to figure out. So I think it starts with these random sequences, and then eventually with more and more infections, you have enough sequences to kind of make it very, very specific to a particular, or make it very, very generic, not specific to different viruses.
Maybe from a single phage, it takes sequences from different regions.
It could do that actually, yeah. It could, in principle, it could do that, right? Because when you're chopping this off, you can incorporate multiple different spacer sequences. But then it becomes an optimization problem of how much do you want to keep into memory versus how much do you want to sort of, do you want to incorporate the entire DNA fragment, or do you want to keep copies of it and so on? Okay, so how does it work, right? Like, so that was the basic discovery. Like, this is what they discovered actually in bacteria. How did they think about applying this as a molecular tool for genome editing? So the genome editing tool uses very similar idea. Take a Cas9 protein, take a guide sequence. This guide sequence will complement to your double-stranded DNA sequence. The moment this starts to hybridize, this Cas9 will start to make cuts, double-strand cuts. These cuts can then be removed or can be repaired using non-homologous end joining. In the process, you can end up including insertions or including deletions. Or in the other way, if you do an HDR, not very important, but if you have another template, you can use or force that template to also get incorporated into this gene. Most use cases will involve just NHEJ, which is just incorporating an insertion, deletion. In a way, you don't have control over this step, right? So this is going to be stochastic in a way, and we'll see what that stochasticity leads to. Okay, so this is overall editing, right? This is editing a particular fraction of the genome so that you end up making lesser amount of a protein. But if I were to carry out, let's say if I have to do a thing in sickle cell, right? So does anyone remember what is the problem in sickle cell? What is the causing?
Single nucleotide.
Exactly, there's exactly one nucleotide which you need to edit, right? So if you start doing this, there is no guarantee that you will end up curing it, right? Because you're either incorporating an insertion or a deletion. You need a much more exact mechanism to be able to do this, and that's what prime editing enabled. So prime editing enables making only single nucleotide changes, single or double nucleotide changes or few nucleotide changes into your region of interest. The idea is very similar. It just uses a smart strategy. So it uses a prime editing guide RNA, which is called PEG RNA. This PEG RNA, which you see here, this entire sequence is PEG RNA, and this is your guide sequence. It's a combination which allows you to identify the target nucleotide sequence. So this is what this particular thing will enable. And then it also has a mechanism to enable encoding new genetic information. And we'll see the example in just the next slide how it enables, right? So it is basically a single guide RNA or your guide sequence RNA along with a primer binding site, which has a reverse transcriptase template sequence attached to it. Now, how does it enable you to make specific edits? You start from this PEG RNA, again, your guide RNA being reverse complementary to the sequence of interest. You attach a particular, whatever mismatch positions you have, you can fine tune it to figure out what particular base changes you require here, right? So for example, in sickle cell anemia, I know I have to make a particular change at a particular location. I will encode this in this PEG RNA sequence itself. The single guide RNA will attach, cross hybridize to it. And the moment this happens, it will form like this kind of a loop kind of a structure. So the 3', so there will be 2 cuts that will be made again, right? So in this, it will, the Cas9, right? The Cas9 protein here, which was doing the double stranded cuts previously, now acts more like a nicking mechanism. So it's going to just make one cut. So it will make a cut particularly at this end, right? So this is the single stranded cut that it is making. So the difference here is not going to make the 2 cuts that it was making. Now it's acting as a nickase and not as an endonuclease, which makes both the strands go away. So with this single stranded cut, this particular sequence will now loop over, will cross hybridize, and it can now make us, so the sequence is designed so that you have complementary sequences which will first recognize this sequence, right? So this is where the first hybridization happened. The nick happened here, so the nick or the cut was made here. This allows the entire rest of the stuff to come and start making a loop to the next strand, to the below strand, right? Then you will perform an RT or a reverse transcription, right? And remember, because the way this is designed, so the reverse transcription mechanism comes attached to this particular PEG RNA sequence, right? So it's extremely complicated in that fashion. But because you have the RT attached here, it will it starts seeing the cross complementarity here. It will start to synthesize this strand. Because of the fact that you introduce this sequence with mismatches here, these 2 sequences or whatever this single nucleotide change you want to get incorporated will get incorporated into this new DNA sequence that is being synthesized. So you will end up making two different copies of sequence, right? So one of them would probably look something like this, where you take this sequence and you start to cross hybridize it, or sorry, make a reverse transcript copy of it, and then your repair machinery will come and it will start to sort of repair rest of the stuff. You will end up making a overhanging string like this here, right? So this strand that was extended will become like an overhanging sequence here. You can have a ligase, so ligases will end up coming and sort of nicking this off in a way, or sort of sewing this up, which will end up to result in a fragment which would look like this. Like, so this fragment unfortunately has no edits. So it is exactly the copy that you had previously, but it is also possible that you get a copy of the fragment coming from the other sequence, which looks like this, where the nick happened such that the RT, after the RT, the hybridization happened with the first strand, but from the other direction there is an overhang. There is sort of a strand which was not sort of ligated. This ligase now will come, will cut this strand, ligate this particular part of the DNA together, and now you have a new DNA which has those edits incorporated, right? So extremely complicated. But what it enables you to do now is incorporate these edits, single nucleotide edits in the entire DNA, by just leveraging this idea that the single guide RNA reverse complements followed by a looping, which allows it to make one cut only. That one cut allows it to form a loop like this. The repair machinery, or the RT machinery will now come make a different copy of this particular strand of the RNA of the DNA. There are two possibilities from there on, that either this strand is already reverse complementary to the rest, to the strand that was not nicked, this remains constant, and eventually some ligation happens and it becomes part of your second strand. In the second mechanism, you end up just synthesizing the original strand copy of the RNA of the DNA as such, right? So the goal in case of single cell anemia is to sort of make it very specific, so that you end up maximizing the chance of this particular thing happening more. Okay, so there are different ways of using CRISPR right now. So we looked at prime editing. It's again repeated here. The first thing that it can do is it can knock out a particular gene, because you perform a double strand break or a double strand repair, which ends up incorporating these indels. You end up making either not making the protein or making a slightly different protein.
This is non-homologous joining.
This is NHEJ. This is non-homologous end joining. Yes. The second thing that you can do, I showed you an example of prime editing, but you can also do this in a slightly different fashion, where you can make single C to T cuts, in the sense that there's a particular if you can re-engineer the Cas9. So most of the innovation that happens in this field is what kind of engineering can you do with Cas9, because that's the cutting machinery. The rest of the principle remains mostly the same. So in this case, this particular machinery allows you to make edits such that the C can be edited only, like it can, if you want to make an edit only if it is a particular C to a T, this particular base editing mechanism allows it. This is very specific to a C to T edit. Whereas the prime editing is more generic. It allows you to incorporate any particular edit at any particular location at the single nucleotide level, right? So how is the second mechanism acting? It's acting by making use of re-engineering the Cas9, so that you also attach what's called a UGI, or a glycolase, a uracil glycolase inhibitor. This will inhibit any sort of excision that happens, except if it is in the proximity to a C. So only editing that is allowed is at the C sites and not at the other site. So a C to G edit, or a C to G or a T to A reverse complementary edits are allowed, but no edits are allowed because you have an inhibitor sequences attached here. Okay. You can also use this to make some sort of epigenome editing, right? So again, if you re-engineer your Cas9, you deactivate it. So this is called a dead endonuclease. This is called dCas9. With dCas9, if you attach it with DNMT3A, which you recall is the de novo methylation enzyme, along with the CRAB domain, we'll see just example of CRAB in just a bit. What this allows you to do is it allows you to make edits so that you can end up depositing methylation marks near the CPG sites or near the promoter site. So you can design your guide RNA to be close to promoter. You can re-engineer of your Cas9 so that the Cas9's property of making the cuts is disabled. So that's why it's disabled or it's dead. It's dCas9. It's not going to explicitly make the cuts. It is going to simply hybridize here, and the DNMT3A will go and just deposit the methylation marks there, right? So this allows you to specifically deposit methylation marks or any sort of histone marks also. You can just change the protein at a particular site without making any sort of NHEJ cuts because your dCas9 has been disabled.
Is this happening on the open DNA or?
Good question. So this requires some recoiling, some uncoiling to happen. So that's why it's near the promoter sequences, which is just generally going to be open DNA. You can also use for something which is called CRISPR-I, CRISPR interference, where again you're not going to make NHEJs, you're not going to make any cuts. You start from a dCas9 and attach a particular protein which is called a CRAB protein. This is associated with Kruppel associated box. What this box of protein does is if you are close to a particular promoter sequence, it's going to inhibit any particular binding to happen of the transcription factors. So it is in some ways it's actually causing some sort of a steric hindrance or some sort of blocking of the promoter, so that without explicitly making any edits or any cuts, you're able to promote or you're able to sort of shut down the gene. Okay. So the difference here is in the interference, you are just shutting out the expression of the gene. In the case of editing or knockout, you're explicitly editing out the DNA, okay? So that you end up making something completely different or not making the protein at all. Whereas here you're just suppressing the expression, RNA expression of that particular gene. Interestingly, you can also re-engineer it to activate something which is otherwise repressed, right? So rather than attaching a CRAB domain, if you attach something called VP64, and if your gene was originally not expressed, this machinery, if you put it next to a promoter which was not very highly expressed, this is able to recruit other protein complexes which allow production of the RNA. So the RNA production, which was earlier low, will now be higher if you do, so this is called CRISPR-I. This is called CRISPR-A. It uses, the only primary difference is the different protein that gets attached. This uses CRAB, this uses VP64. This activates, this represses. Okay, is that difference clear? So it's not just about editing, it's actually also about re-engineering in the sense that you can do an epigenome editing. So you're not explicitly editing out the DNA, you're not making any DNA changes, but you're still able to control the RNA expression. So it's used a lot in the field.
How long can this stay?
Good question, very good question. It can, so that's a very system dependent thing, and it's a function of how much amount of thing you add. I actually don't know the numbers at the top of my head. But if you add, it's mostly driven by the amount of Cas9 protein that you engineer in the cell, right? So the more amount of it is, actually the way we'll see this in the, so let me answer this in a bit. So when you do this kind of editing, right, you're also making the cells compete in some fashion, so that if you have to measure the phenotype, you have to measure what changes exactly happened. So you will do this perturbation, but you'll put them through some sort of a competition where you're allowed, where you can measure what changes happened. So this can range all the way from few days to from a few 1 day to like 7 days. I think that's the general trend in which people do these experiments on.
It doesn't allow DNA replication.
Doesn't for these interferences, the epigenomic editing?
Yeah.
So this, at this point, it is only sort of acting. So it is attaching at the DNA, like the complementation is happening at the DNA. But the machinery that it is affecting is the Pol2 machinery overall. Like this is suppressing it, this is activating it, and this just because you're doing the deposition of the methylation marks is going to suppress it. So the action is happening at the RNA level, but the things are acting at the DNA level. Like the complementary sequences are at the DNA level.
No, does it inhibit the DNA polymerase activity?
Oh, does it inhibit the DNA polymerase activity? No, not directly. Not that I know of in terms of what happens. Because
it's binding to the DNA sequence.
It is
both the strands.
Both the strands are complementary. So this strand is complementary to the DNA sequence. That's right. Yeah. So it doesn't affect directly your DNA part.
Can you go to one of the previous things?
Yeah.
So this is how it looks like, right? So it's binding to one other strand.
It is. At least one of the strands, yeah. So it depends on, like, for example, in this case, your this will attach to one of the strands, and then it will.
And DNA polymerase needs to attach to both of the strands.
So it has to unwind first. Like the DNA poly 2, when it comes, it needs to unwind first before
Yeah.
it starts. So that needs to be unbounded DNA for it to
And
start.
so
doing
does
that.
this affect the unwinding?
Oh, good question.
It should, right?
It should, in a way, yeah. I think. Good question.
You
I-
said that this is persistent for 7 days, but I would imagine that the cell replicates at least a few times.
Yeah, of course. So that is happening in parallel. That's a good question. I should look it up. I don't know at the top of my head how do they interact. Do you know?
The Cas9 protein gene is itself incorporated in the Cas9 gene is incorporated.
Yeah, you're re-engineering the cells to produce the Cas9. His question is.
Affect the replication.
Okay, so as soon as the cell replicates, it makes new Cas9 proteins and then it goes back to inhibitor.
But you have the Cas9 already alive, right? Like it's not dying as such. So there is some Cas9 production that you already have. So his question is, if something is going back to the DNA and hybridizing, can it suppress the production? Can it suppress the movement of DNA poly 2? Is that what your question is?
Or just one step behind. So before that, when the DNA needs to unwind for DNA polymerase activity to begin, does this inhibit the unwinding of the DNA? Or does it just get removed easily when you want to?
I get your question, but since the Cas9 is itself inserted into the gene and still normal replication occurs, so I don't know exactly what occurs, but the replication does occur because cells.
Replication does occur. Yeah, I think that's true. I think his question is more about the
This probably
amount.
gets removed in some mechanism before DNA replication occurs, and so probably it gets removed during or before the
So
unwinding.
it doesn't get removed because it can persist actually, like in terms of you can, since it's sort of a loop in a way, right? So you're engineering the DNA itself to produce, you're engineering the cells itself to produce the Cas9. My interpretation of your question was, does it affect the replication machinery at the DNA level itself, right? So my intuition is not, but I will double check in terms of, is your intuition the same in terms of what happens?
And now that he has said that it doesn't affect the replication, what I think is happening is that it right now when it's staying at the DNA, right? So it's binding to one of the strands. When the DNA starts unwinding, this gets removed from that one strand itself also. So it lets the DNA be free for some time, and then
Yeah, in terms of
comes it replicates it, and then after the cell duplicates, when it starts creating proteins again, it again creates this Cas9 protein.
Yeah, so there is a delay in terms of Cas9 because it needs to get replicated first into a different copy before it
Yes.
actually gets inside. Fair enough. That's fair enough. Okay. In terms of other things that it can do, it can also be used to do some sort of RNA modulation, which is that you can modulate, do some sort of RNA base editing using CRISPR. So this is sort of still in a way a very much upcoming field. One of the applications of this is if I want to, for example, do a base editing at RNA. So similarly, using the principles of just CRISPR base editing, rather than at the DNA, I can do this directly at the RNA level. So the hybridization here is happening at the RNA level. You can do this by incorporating what's called ADAR, or which is an adenosine deaminase, which is targeted, which specifically targets RNA. It converts your A's to C's or A's to I's, and then that gets converted into G's eventually, and that will end up making, so you end up starting with a particular base in your guide RNA, which is C. So if you want to make base editing, that is also sometimes people do it.
This is single base editing.
This is single base editing at the RNA level. Then you can also do similar sort of, depending on which kind of complexes you add, you can also do modulation at the splicing level, right? So this is about where the RBFOXN and RBFOX1C play a role. So they can, what you're doing essentially is you're changing again the Cas9 to re-engineer it so that you can attach these particular RNA binding proteins. So that they reverse complement, and these proteins are involved in these are not RNA binding. These are proteins where the boundary of that protein is now cross hybridizing or in a way attaching to the Cas9, And this plays a role in how the splicing happens. So this allows you to splice out specific intronic sequences if you design your guide RNA to be targeted along that. So what will happen essentially, it will cross hybridize to this intron, you have the splicing machinery, this will get spliced out, you will end up joining these two exons. And then you can use this to again perform RNA interference. So this is very, very similar, which we already talked about previously. Okay, now how would you do this, right? So this is at the gene level. How would you do this in a high throughput fashion? So this question that we asked about being able to understand what the function of each gene is, how would you do this? What kind of experiment would you have to do?
For single gene, what are we doing?
For single gene, we can do CRISPR. We have to design a guide RNA against that gene. How would you do it for?
To identify activity for a single gene, is that is why we use CRISPR, right? So now we want to modify multiple genes.
Yeah.
So change the guide and have multiple.
Yeah, so you'll have multiple guide RNAs, and this is what the entire field of CRISPR screens does, right? So there are two broad fields, which is pooled and arrayed CRISPR screens. So in pooled CRISPR screens, what you do is you re-engineer the cells so that your cells can express Cas9. Cas9 is again the protein that you really require. And then you come up with different guide RNAs. So you make a library of these different guide RNAs so that they can transfect or they can affect or they can cross hybridize or do some sort of hybridization with your genes of interest. So I will ideally would have multiple different guide RNAs for each gene because the editing is not exact. So if I have only one guide RNA for one gene, it might end up doing it. It might end up not making the edits. So if I have like 4 or 5 copies of gRNAs, guide RNAs for each different gene, I can take a virus. These are called lentiviruses. This can invade my normal growing cells these are human cells, could be any animal cell. I re-engineer them. So I've re-engineered these cells to produce Cas9. The guide RNA, I'm delivering it from outside using the lentivirus. The lentivirus will go deposit the guide RNA sequence to it. Now, this guide RNA will cross hybridize. You have the Cas9 attached to it in a way once it goes inside. Now you will ask the cells to compete, right? So you will either do some sort of a drug treatment or you will do some sort of a measurement which will allow the cells to, you're essentially checking the survivability of the cell. Like that's the phenotype that you're testing them against basically. And that's what finally you end up retaining. So what is the screen for? The screen is for.
So the only test you're doing is the growth rate.
Is the growth rate in a way, yes. So that is your phenotype that you're measuring here in the sense that what happened to the total number of cells, how did they survive? And in the process, what happened to, if I did a enrichment, if a particular cell got a particular guide, what genes change in the process? Because obviously the cells that you're measuring, they have survived. So they've survived, so what changes happened in those cells? You could do this in 2 different ways. You could either do this in like this particular fashion, which is that you take the entire library, you just go berserk, you just transfect everything with all possibilities. You can do it in a more structured way. We can, so this is when you're exploring things, when you're figuring out what changes happen to what genes and how do the changes get incorporated in the cell. The arrayed CRISPR screens are more directed in the sense that rather than sort of perturbing each different cell with each different guide, you take a much more structured approach, is that you will arrange them in wells and you will give them, you'll give certain pool of these cells a certain guide and then eventually do the same thing, which is you will make them compete. You can make them compete with different drugs and then come back and ask what is the impact given a particular drug that I treated this with and a particular knockout of a particular gene, what is the impact on the rest of the genes that it did, right? So this allows you, this is more sort of discovery based where you don't have any idea about what is going to happen. Here you have already narrowed down a few genes of interest and you're doing this in a much more structured way.
So there are different drug treatments in different cells.
So yeah, it really depends on the context that we are asking this question in, right? So in principle, if I'm asking this question whether a particular gene does good in cancer versus not, I will do some sort of treatment which I will take the cancer cells versus the normal cells and see what impact knocking out a particular gene has.
So in the array treatment, if I add two guide RNAs in the same cell, it still doesn't guarantee that all the cells.
That's right. That's a good question to figure out. That's a question you sort of actually most people don't use array.
If we take that 50-50 figure from CRISPR, then only 25% will have both.
That's right.
Both of them.
That's right. So and also this is somewhat stochastic, so there's no guarantee that you'll actually end up getting.
It keeps getting worse, right? So now if we have 3 guides, then it's only 12%.
That's right. So that does involve some amount of math to be done in terms of how many guides should you use to guarantee a certain kind of infection rate and so on. Okay, yeah.
Related question, how big is your library?
How big is my library? Yeah, good question. So this people perturb all the way from, so this is called perturbation. You're perturbing the gene, right? So it depends on, at this point it's more of a function of how much money you have, right? So if you have a lot of money, you will develop what's called genome-wide perturbation screens. So you'll just perturb every possible gene in the world. In the 20,000. Yeah. So there is a library. This came out from a lab. Jonathan Weisman, I think he's at Whitehead Institute, which is affiliate. I think it's MIT for all practical reasons. So this was done in 2022, where they perturbed every possible gene in a particular cell line and figured out what kind of changes happen overall.
Single perturbations or?
I think they use 3 guides per RNA.
Pooled array?
Array. Sorry, pooled. This is all pooled. This is all pooled CRISPR screens.
So
Yeah.
controlling.
There's no control. Yeah. You're looking for what changes happen once you perturb them in the sense. Okay, what does it involve, right? So the first thing that if you want to do these kind of experiments is you have to design a guide RNA, and this guide RNA design, there are softwares to do it. The first part of the course, ideally, I wanted this course to end by us developing some sort of a guide RNA design tool. Unfortunately, we'll not be able to reach there, but you're doing projects, so you're kind of doing something related. So we have to design a guide RNA which is 19 to 21 nucleotides long. It is to be designed such that what makes it so specific, right? It has to be adjacent to a PAM, to a protospacer adjacent motif. What are these proto-adjacent motif? These are generally in the form of sequences like NGG. I will go back just because I realize I did not emphasize this enough. So these sites where the guide RNAs get incorporated, right? There is a special property that they should have NGG, or this N stands for any base followed by a GG, or an NAG, any base followed by AG. This is just because the cuts are enabled. So the nuclease cuts that it is doing, right? The nuclease cut that Cas9 is doing, it recognizes this motifs particularly. So whenever you're designing guide RNA, you need to be adjacent to these 2 particular sites. Okay, so let me go back now. Okay, so in terms of how many sites, how many NGGs will occur? I wanted this to be a quiz actually, but the answer is already there, right? So for every 8 nucleotides, you will see a NGG kind of a sequence occur. So it's not a very difficult, like the first time you see GG, I have to always think about designing something which is next to a GG. You will think that
NGG and NAG both.
Yeah, NAGG both. Yeah, so this is, but the probability is the same, right? This is 4 sequence. You can combine it, sure. You can.
Every 8 is for either NGG or NAG, or?
Yeah, no, it is either NAG or NGG. Yeah.
Isliye 2 ka factor hai?
No, 2 ka factor is because it's a double-stranded DNA.
So it can occur in either of the strands.
Either of the strands, exactly. So this, I will sort of clarify this, but this is NGG specifically for NGG half 1/4, 1/4. This is 1/4, 1/4 times 2 because there are 2 strands of the DNA. So every 8 nucleotides is where you will see the change.
So if the endonuclease detects NGG in either of the strands, it will cut both the strands upstream.
Yes, so it has to. So when the site sgRNA reverse complements to it, there has to be an adjacent motif to it, right? So and then it will make a cut here, and it will make a cut to the next 5' side of the things. So it will make 2 cuts, so that it makes.
So on either strand, it's fine.
On either side is fine. Yeah, it doesn't need to be on both the strands. On either strand is fine. Okay, So fine, so that is doable. In terms of the spread, so we'll not be doing any particular data analysis for these particular screens, though it's a very interesting, very interesting problem. We'll not be able to do it just because we have other things to cover in the next 3 classes. Maybe one of the last, so the 3 lectures that I'm going to design to be online will be more on the hands-on exercises, because I realize hands-on is anyway you can just do it on a recording basis. So you'll do that maybe you'll do one of the exercises as a hands-on exercise there. But the idea behind analysis is very simple, right? So if you have any sort of conditions, you infect a particular gene, you design multiple guide RNAs against it, you will have to quantify, right? So one thing that I did not focus on is, which I'm going to right now ask you, is how will you capture these guide RNAs? This is just the last slide, so we'll just end it in 2 minutes. But how will you capture this? How will you capture the guide RNAs? You also would like to sequence them, right? Otherwise, how would you know whether the infection happened or not? I can sequence the rest of the RNA because they have a poly A tail to it. But I also want to, because it is, whatever I'm infecting my cells with, I need to know what infection it got, right? So here, when I took the library, when I infected the cells, I also want to know what infection did it get, which guide RNA did it get. I can pool the RNA, I can get the RNA content of this cell, that's a solved problem, right? We can do single cell sequencing, we can get it. But how do we get the guide RNA? That'll make it slightly complicated, but yeah, that's one way to do it. Sure, you could do some sort of.
I think that it's more simple.
More simple? No, it's harder if you want to do it in a high throughput fashion. Any ideas? How would you? Yeah.
What did he suggest?
What?
What did he suggest?
He suggested radioisotopes. So some sort of like from that I'm interpreting some sort of signaling that you're doing basically, right? Some sort of fluorescent signal or something like that. Yeah. Any other ideas? You want to capture the guide RNA also. Fluorescence is similar to what he said, but yeah, that will be one way. But think about like 20,000 genes, like 20,000 fluorescence, hard, very hard. There are not that many channels.
And computational, I mean, you sequence everything and search everything.
So you could do that, but yeah, I'm actually not doing a good job because I'm not telling you what the problem is. So the problem is this, right? That RNA has a poly A tail, so we have a mechanism to capture it. Guide RNAs don't have it. There's no mechanism for it to capture. We don't have that mechanism, right? So when this infection happens, right, when this thing gets incorporated, this guide RNA is there, like it is there, it's there somewhere, essentially in the cell, right? We want to be able to capture it. If it had a poly A tail, we would have been able to capture it because it would just be around.
Would this show up in sequencing?
It will show up. It will show up in sequencing, yes. But you have to stabilize it a bit, yeah. You could do PCR, for example. Yeah, you could do PCR for a particular, but again, if you want to do it for like 1 million cells, very hard to take one cell and do PCR, right? But yeah, very similar principles. What you can do, and I did not highlight this here, when we do the hands-on, maybe I'll highlight it there, is to sort of engineer a vector so that when you're delivering the guide RNA, that vector has the rest of the mechanisms figured out, which is that it has a poly A tail somehow, so that you can sequence the guide RNA separately. You can figure out if it is present in the cell, you can use the entire strategy that you use for capturing poly A's to also capture the guide RNAs. Okay, so briefly the steps are you sequence them, you quantify them, and then you eventually figure out what genes are changing, right? So what genes are changing between these two conditions, given that, or you're perturbing certain genes, but you want to figure out what is the impact that those genes had on something else, right? So you would want to perturb something like transcription factors, for example, particularly if you're doing this kind of experiment, to understand what other genes is it affecting. So this will give you a causality that if I change this gene, this is the impact that happens on the
number of reads, right?
There's a number of reads, yes, that's right. Okay, so there are a lot of applications. Like this is what has enabled some of the like this is truly translational in the form that once it works, and once you figure out what a particular function of the gene is, you can use this technology to figure out what impact it is having on the expression, what impact it, or particularly in case of applications to immunology, cancer, what impact a particular mutation has. This is the way to figure out that if I'm making a claim that this mutation causes cancer, I will go come and do a base editing of that particular mutation and show that the cell that I have is now growing at a much faster rate. Like that is what, this is the golden truth behind any claim that a paper makes. So if I make a claim that gene A does X, I can go do a CRISPR on gene A and show that okay, X does happen if I CRISPR it out or CRISPR it in. Okay. Okay, so we'll end it here. Any questions? Okay, thank you. So see you next week. So next week also we'll continue on some of the themes of the course. Particularly want to just introduce what is really multiomics. And then the session after 30th will be hands-on. So there'll be 3 classes, all hands-on, pre-recorded. You will have access to templates. You can play around. Please, if you have not started working on the projects, please do. The assignment 7 will also involve questions from your assignment. I'll go through your first questions. The next week I'll have to see that you're making progress, right? So the second question that you get will be conditional on what you've shown for the first. So take it seriously. We only have 3 weeks remaining, but I think that's sufficient time for wrapping up. Also, if you have any questions related to project, please schedule a meeting if you would like me to give you feedback.